Hello, everyone, and welcome to Advancing Precision Medicine in Inflammatory Diseases with Affinity-Based Proteomics. I'm Matt Thorne of LabRoots, and I'll be your host for today's event. Today's educational web seminar is brought to you by Olink and presented by LabRoots. To learn more about our sponsor, please visit olink.com. We do encourage you to participate today by submitting any questions you have during the presentation. To do so, you can simply type those into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You can also submit any technical issues there as well if you're having trouble seeing or hearing the presentation. I'd now like to welcome Valentina Menegin, Global Marketing Manager at Olink.
Thank you, Matt, and hello everyone, and welcome to another Olink Multiomics coffee break, this time organized in collaboration with Cobiomic and LabRoots. I'm Vale Meneghin, Global Marketing Manager here at Olink, and I'm pleased to give you a quick overview on Olink Proteomics for those of you who are new to our company and technology. Okay, so Olink is a Swedish company by origin that in a few years has grown rapidly from a small Sweden-based company offering, say, for a few hundred proteins to a global leader with a broad portfolio of flexible protein biomarker solution and a library of high-quality, thoroughly validated assays that cover more than 5,000 proteins. Our customers continue to drive significant advancements in the field with over 2,000 publications to date, covering a broad range of applications and disease areas. And as you may have heard, we are really proud that the UK Biobank Pharma Proteomics project chose the Olink Explore HD to measure over 5,400 proteins in 600,000 samples from the UKB participants. And this is the largest ongoing proteomics study that aims to create the most comprehensive map of protein levels in the human body, significantly advancing our understanding of disease, which is indeed in line with the Olink ambitious goal of revealing the truth of human disease protein by protein. And we are not alone in this mission. We are doing this together with our customers, the research community, and with a strong network of certified service providers. And we are really proud to present to you the Olink CSP network that is equipped to honor the Olink commitment of delivering excellent data to our global research community. The Olink CSPs are trained by Olink scientists and undergo annual recertification through concordance testing. And for this reason, they can help you to accelerate your research, avoiding any roadblocks and facilitating the integration of the cutting-edge proteomic solutions into your studies. And we are really proud that Cobiomic is part of this prestigious network, as this ensures that you can confidently outsource your projects to them, whether you are working on low or high throughput studies. And before moving on to the agenda, we are excited to share our Olink mouse cytokine grant program today with you. This is organized in collaboration with Olink certified service providers and, of course, with Cobiomic. And this is an excellent opportunity for those of you performing mouse immune protein biomarker research to get started with Olink. So you just have to submit an abstract on how your study can utilize this panel to capture novel translational insights, and you will get a chance to win a free pilot project. An internal scientific committee will choose winner based on the research creativity and innovation. So I suggest you to follow the QR code links for your region and register to the grant program, as the deadline will be the 16th of April. And now, without further ado, let's move on to the agenda. So today I'm pleased to welcome Dr. Nuria Barbarroja and Dr. Carlos Perez Sanchez from Cobiomic, who will present how they can support implementing Olink in your research studies. And they will also present one of their recent projects showcasing how the reverse translation of human proteomics insights into mouse models can enhance our understanding of molecular pathways and drive the development of effective therapeutic intervention. So, Carlos, over to you.
Thank you, Valentina, for the introduction. So, hi everyone. I would like to spend the next 2 or 3 minutes talking about cobiomic in order to show you what we are doing and how we can support you. Okay. So, the first thing that I would like to say is that cobiomic is an spin-off of the University of Córdoba, and also is an spin-off of the Biomedical Institute of Córdoba. So at the end, we are like an academic company that basing our research, our result, we decided to create this company in order to bring new tools that can improve the life of many different patients. Okay, this is the main goal of cobiomic. Here I'm showing you the team. So we are a multidisciplinary team integrated by principal investigator of different research groups, by rheumatologists, by scientists, also by informatics. So we have more than 15 years of experience in the field of researching about inflammatory chronic diseases. We have more than 500, more than 100 publications, 5 patents, and different awards. We have 2 main areas in our activity. So we have one part of the company in which we are developing precision medicine tools for rheumatic diseases, but at the same time, we are also providing services using Olink technology. So you can see that both parts of the company are overlapping because we are using the Olink technology for our own research, but at the same time to provide services to other researchers. So regarding the first part, the R&D area of our company, as I said, we are developing a blood test that is able to predict if patients with rheumatic diseases are going to respond or not to different therapies. So we use a combination of proteomic data with clinical data of this patient, and then we generate machine learning algorithms that can provide a report about the probability to respond to a particular therapy. Regarding the service part of our company, we were the first certified lab by Olink in Spain in 2022. And we are like quite unique in many different ways. For example, as I said, most of us are academics, so we are researchers. We are providing services, but at the same time, we are researching with this technology. We have published more than 10 papers using Olink technology in the last 2 years. We have other characteristics. For example, we deliver the data only in 7 days. So we are fast in this way. We have also expertise in alternative matrices. So most of the samples that we receive are serum or plasma, but we are also working with many different alternative matrices, such as urine, tears, saliva, and samples like that. We have analyzed more than 12,000 samples. We are working with more than 60 organizations so far. Here you can see the main panels that we are doing with our customers. So we are providing different formats of Olink, such as Target 96, Target 48, Flex, and now Reveal. Here you can see that most of the researchers are using the panels related to inflammation and cardiovascular disease, among others. The area of research that we are supporting, you can see here also that most of them are related to cancer, cardiology, neurology, and rheumatic diseases. And we are also working with pharmaceutical companies. So we are doing projects for example, Lilly, UCB, Almiral, and another biotech companies, because we are supporting with Olink technology the drug development, drug discovery, cosmetic, biological age, and the diagnostic. Finally, I would like to also show you some national research centers that we are working with. Also, we are providing services to international research centers. And also, as I said before, we are working right now with pharma and biotech company. Here you can see some recognition that we obtained in the last year, more than 15 awards in the last year, thanks to the activity that we are doing. We are happy to support your research, and I hope that you can enjoy the webinar with my colleague Nuria. So Nuria.
Okay, hello everyone, and thank you for joining this webinar. I hope you find the session insightful and worthwhile. I would like to extend my gratitude to Olin for the opportunity to present our results in our research, including the application of Olin technology. One of my research areas is focused on the study of a chronic inflammatory disease that primarily affects the joints and the skin, known as psoriatic arthritis. But beyond joint inflammation, we need to consider the increased cardiovascular risk in these patients, since cardiovascular disease accounts for up to 40% of morbidity in this population. This risk is driven by systemic inflammation, but also by obesity, metabolic syndrome, and even the effects of treatment and or lifestyle factors. So the treatment approach shouldn't be just aimed to reduce inflammation or control psoriasis, but must also address cardiovascular risk as a key therapeutic target.
Sorry, Nuria, for interruption, but
I can move the presentation. The animation.
Yeah, Nuria, we cannot see the slides moving. So if you can click on next.
Oh, no. Yes.
Okay, great. Thank you.
I'm sorry. Now, okay. Okay. So in this sense, apremilast is a treatment commonly used in routine clinical practice for psoriatic arthritis. It is a phosphodiesterase 4 inhibitor that modulates immune response by increases intracellular CMP levels. This leads to a reduction in pro-inflammatory cytokines and an increase in anti-inflammatory mediators. But PDE enzymes also regulate metabolic and endocrine pathways. Since CMP plays a key role in adipocyte differentiation and lipid metabolism, apremilast could potentially influence the cardiometabolic health. So the question is, could apremilast reduce cardiovascular risk in patients with psoriatic arthritis beyond its inflammatory effects? In this study, we explored the cardiometabolic profile of patients with psoriatic arthritis, combining clinical and molecular data. So in our core, we observed a high prevalence of metabolic abnormalities among patients with psoriatic arthritis, including insulin resistance, obesity, hypertension, and dyslipidemia. When we look more closely, we saw that patients with insulin resistance or obesity had more swollen joints, higher disease activity, and elevated inflammatory markers like C-reactive protein and erythrocyte sedimentation rate. So metabolic dysfunction doesn't just coexist with the disease, it actually makes the disease worse. We use Olin Target 96 cardiovascular panel to analyze the plasma proteome in patients with psoriatic arthritis. Several proteins related to inflammation, lipid metabolism, vascular biology were significantly dysregulated. The protein interaction network shows a strong functional clustering in immune and inflammatory pathways, lipid metabolic processes, leukocyte migration, and adipokine signaling. Altogether, this data reveal a specific proteomic signature in psoriatic arthritis that could contribute to cardiovascular risk and potentially be targeted in future interventions. So we found elevated levels of several biomarkers associated with metabolic comorbidities and clinical details of the disease, such as insulin resistance and obesity, disease activity and inflammation, body surface area with psoriasis and onicopathy. And among all the proteins, CD163 was commonly associated with metabolic comorbidities, disease activity, and clinical details, suggesting a key role of this molecule on the pathogenesis of psoriatic arthritis. We then conducted a longitudinal study with 45 psoriatic arthritis patients, 15 treated with apremilast immunotherapy, 15 with methotrexate, a mainstream treatment used also in psoriatic arthritis, and 15 with the combination of both. Okay, Patients were classified into 2 clinical clusters based on their metabolic profile. So cluster 1 had a low prevalence of metabolic comorbidities, while cluster 2 had a high burden of alterations such as obesity, insulin resistance, and atherogenic risk. We then analyzed the circulating proteome and we found different patterns in each group. Cluster 1 showed minimal changes compared to healthy donors, while cluster 2 had a strong upregulation of protein related to inflammation, lipid metabolism, and vascular dysfunction. We then analyzed the response to treatment in both clinical clusters. In patients without metabolic comorbidities, cluster 1, all the treatment reduced the disease activity, BMI, and insulin resistance, with a slightly greater effect seen with apremilast. In cluster 2, which included patients with metabolic alteration, apremilast was particularly effective in lowering the activity of the disease, BMI, and HOMA-IR after 6 months. Looking at the molecular level, we saw that apremilast, alone or in combination, reversed the expression of several cardiovascular-related proteins, especially in cluster 2. So these results suggest that apremilast may have a dual benefit, improving both inflammatory and cardiometabolic parameters in psoriatic arthritis. So from this clinical study, we identified distinct clinical and molecular profiles among patients, showing that metabolic comorbidities are associated with higher disease activity and altered cardiovascular proteomic signature. Importantly, we observed that apremilast not only improves inflammatory outcomes, but also positively modulates metabolic parameters and cardiovascular related proteins, especially in patients with a high cardiometabolic burden. And these findings support a more integrative approach to psoriatic arthritis management and open the door to tailored treatment strategies targeting both inflammation and cardiometabolic risk to optimize patient outcomes. So, in patients with psoriatic arthritis, there is also a higher prevalence of MASLD, formerly known as NALD, and now referred as metabolic dysfunction-associated steatotic liver disease. This association involves mechanisms such as obesity, insulin resistance, systemic inflammation, and genetic factors. Given the metabolic and anti-inflammatory action of apremilast, we aim to explore its potential therapeutic effect in this setting. So our objective was to evaluate the effect of apremilast on MASLD using a mouse model. We are happy that this work has been selected for the presentation at the upcoming EULAR Congress in Barcelona as part of a poster tour on molecular treatment signature in psoriatic arthritis. So to study the effect of apremilast on MASLD, we used the GAN model, an established mouse model of metabolic dysfunction. These mice were fed a high fructose and high cholesterol diet for several weeks, leading to obesity, insulin resistance, and liver steatosis, closely mimicking human metabolic disease. After disease induction, mice were treated either with vehicle or apremilast, and metabolic tissues, including liver, different adipose depot, and skeletal muscle were collected for analysis. Wait, something was wrong with this slide. Okay. Let me check. Okay, so in this slide, I will tell you what was in this slide. As expected, the GAN diet successfully induced key feature and metabolic dysfunction in mice. So we observed a progressive weight gain and increased fat mass together with elevated levels of insulin and HOMA-IR score indicating insulin resistance. In addition, we also observed hepatic alanine aminotransferase levels that were significantly increased, reflecting liver damage. Now, So apremilase significantly influence body composition in the fed mice, and we observe a clear reduction in total fat mass compared to untreated animals. This effect shows a consistent trend across several adipose tissue depots, particularly in mesenteric fat, and was also evident in the liver. When we then analyze serum biochemical markers to evaluate the systemic metabolic effect of apremilase, while glucose and aspartate aminotransferase levels remain unchanged, we observe a clear reduction in circulating insulin HOMA-IR and ALT in treated mice. Okay. To better understand the tissue-specific effects of apremilase, we perform a proteomic profiling using all-in-target 96 mouse exploratory panel, and we analyze 5 key metabolic tissues: epidermal, mesenteric, and inguinal adipose tissue, skeletal muscle, and liver. This approach allows us to capture the molecular impact of both the GAN diet and apremilase treatment across the different metabolic compartments. We then explore how protein expression in each tissue correlated with key metabolic variables: body weight, insulin HOMA-IR, and ALT. Across all 5 tissues, we identify proteins significantly associated with these parameters, especially those involved in inflammation, lipid metabolism, cell recruitment, proliferation, and tissue remodeling. This analysis helps pinpoint molecular pathways that are most affected by the GAN model and potentially reversed by apremilase depending on the tissue. Now here we show the fold change of all the proteins modulated by the GAN diet across 5 metabolic tissues. As you can see, apremilase treatment partially reverses many of these changes with distinct patterns depending on the tissue. This figure highlights the protein significantly altered across metabolic tissues in response to the GAN diet, and we observe overlapping changes in fat depots, liver, and muscle, despite tissue-specific profiles. Common functional themes emerge, including inflammation, lipid storage dysregulation, and stromal remodeling and fibrosis. This pathway may represent key targets for therapeutic intervention in metabolic dysfunction-associated diseases. We next focus on proteins significantly modulated by apremilase across the metabolic tissues, while apremilase consistently down-regulate key pathological pathways. Its effect was highly tissue-specific. These findings underscore the distinct molecular response to each tissue, highlighting the complex action of apremilase in metabolic disease. So from this mouse study, we can conclude that using the target 96 exploratory mouse panel, we were able to characterize the proteome of several metabolic tissues with a single assay. The GAN diet induced marked changes in the protein expression across all tissues, with epidermal, adipose tissue, and liver being the most affected. Altered proteins were mainly related to inflammation, fibrosis, adipocyte dysfunction, immune cell recruitment, and tissue remodeling. And finally, apremilase modulates protein expression in all the tissues in a tissue-specific manner, with the most pronounced effect observed in the liver. So this is how we translate our clinical findings into a mouse model to explore new effects of a drug already approved for psoriatic arthritis, which could potentially be repurposed for other applications such as MASLD. Needless to say that these findings require further validation and confirmation through clinical trials in humans. So, apart from Cobiomic, this work wouldn't have been possible without the dedication and the collaboration of my amazing research team. Thank you all, and I'm happy to take any questions.
Great. Thank you both very much for your presentations. Before we do get into that live Q&A, we do have a couple of poll questions.
Exactly. So thank you, Matt, for launching the poll question. So we kindly ask you to reply to these questions because this will help us to better plan the upcoming webinars. So the first one, are you currently working on protein biomarker studies? Please reply to this one. We will allow a maximum of 30 seconds. Okay. So next question. If not, are you planning to start in the next 6 months? Okay, next one. So do you want to be contacted by an Olic rep to learn more about the Olink PA technology and how it can be integrated into your current omics workflows? Just let us know if you require more information from us, and we will be happy to contact you afterwards. Another few seconds. On the other side, do you want to be contacted by Cobiomic to start your Olink project? Please let us know. Which applications would you like the next webinar to focus on? And here you have the list: aging, cardiovascular disease, immunological inflammatory disease, neuro, oncology, immuno-oncology, population scale proteogenomics, biomarker discovery. Okay, to clinical utility, proteomics in drug development. Let us know. In the meantime, I remind you that we will now open the Q&A session, so feel free to add all your questions in the Q&A box. Okay, I think that we can conclude the poll questions, Matt, and you can launch the Q&A.
All right, thank you. And like she said, go ahead and put those questions in that Q&A box in the bottom right-hand corner. You'll be able to then get those questions answered. So now I will turn that over to Pilar to start going through the Q&A.
Hi, hello. Thanks, Matt, for sending me this. I'm Pilar. I'm the field application scientist in charge of the Southern Europe team, and I will be taking the Q&A. So, Nuria, maybe you can take these questions. The first one is, what was the detectability of the proteins on each mouse tissue studied?
So in terms of detectability, we exclude, in this case, proteins that were below the limit of detection, the low LOD, in more than 75% of the samples. So the percentage of exclude proteins per tissue was around 15% in liver, in skeletal muscle 18, I remember, and in adipose tissue was around 5%. So the detectability in adipose tissue was higher, but the panel was really good. So good coverage across all the tissues.
Great, thank you. The second question is, why cardiovascular disease 3 panel was chosen for your study instead of cardiovascular disease 2 panel?
This is actually a common question from customers. And we choose, in this case, cardiovascular 3 panels over cardiovascular 2 because it's a specific content. So cardiovascular 2 includes cardiovascular and inflammatory biomarkers with a strong focus on exploratory proteins that may become future markers. And cardiovascular 3 includes cell adhesion molecules which were particularly relevant for our study. So that was the reason.
Great, thank you. Another question, how many proteins do you analyze with the target 48 mouse cytokine panel?
So they are 45 cytokines, and you can measure in one panel 40 samples.
Great. And which type of samples can be profiled with the Olink mouse panel?
So as you saw in my presentation, we can analyze any type of tissue. So through protein extraction, we can include urine, we can include, of course, serum and plasma, and also supernatant of some, you might in the in vitro study with some primary culture from mouse cells or tissue, and you can measure also the supernatant of that experiment. And I don't remember more. I think that most of them you can do it.
Perfect, thank you. So we
Very
have
welcome, Pilar.
we have some other questions, but we will take them offline. So we will reach out to the people who have asked certain questions individually. And for now, this is all. I don't know if you have more questions, but if you do, please let us know. We will be happy to take them.
All right. So before we go, we would like to thank the audience for their time in joining us today. And thank you once again to our speakers for their important research. Thank you to LabRoots and our sponsor, Olink Proteomics, for underwriting today's educational webcast. For any questions that we didn't have time for today, and for any of those that get submitted during the on-demand, you can receive those answers via the information you used when you registered. The webcast will be able to be viewed on demand, and LabRoots will alert you via email when it's available for replay. And we do encourage you to share that email with your colleagues that may have missed the live event today. So until next time, thank you once again, and goodbye.